Hello, welcome back to Father-Offspring Interviews. This is episode 47. We're getting right into it with the question, could you please talk about neurogenesis?
Neurogenesis, the genesis of new neurons. In this case, in the adult brain, because that's where there's been a revolution in the last 25 years or so. Okay, if you took an introductory neuroscience class anytime in the last century, anytime since the Roman Empire, since humans were in the bushes having sex with Neanderthals, you take an intro neuroscience class, and the first thing they teach you is at birth, you've got all the neurons you are ever going to have. You have your maximal number. You don't make new neurons once you're born. Skin, you make new skin all the time. You make white blood cells, lymphocytes when they're making new antibodies. You make new ones. You make new red blood cells. Neurons don't. Ninth grade biology, a cell splits into mitosis. Neurons don't do mitosis. Neurons are post-mitotic. And thus the standard story would be, because neurons are post-mitotic, you've got all the neurons you're ever going to have at the time that you're born. And if you squander them away with senescence or alcohol or imprudent this or that, all you're going to do is be depleted of neurons over time. Once a neuron is lost, it is gone forever. Okay, so weirdly, there's a slight piece of good news with that. You will recall, perhaps from a recent episode, what's cancer about? Cancer is when you accidentally reactivate growth genes that were very useful early in life, and you reactivate them by chance later on, and they suddenly cause cells to start dividing and cause a tumor and all of that. Because neurons are post-mitotic, there's virtually never cancers of neurons. Cancers in the brain are usually of glial cells, glioblastomas. Okay, so that's good news. The bad news, however, far outweighs that, which is you lose a neuron, and by the time you get to a certain age, you're losing a certain number of neurons in certain parts of the brain. They are gone. You can't get them back. And thus the notion that actually the adult brain could generate new neurons, adult neurogenesis, was simply impossible. Except it turned out not to be impossible. And an epic story with this, and nicely I got to have a front row seat to some of it. It all started in the 1960s. A young scientist named Joseph Altman, who was starting off his career at MIT, was using some fancy new techniques at the time and finding evidence that in the hippocampus, part of the brain having to do with learning and memory, that the hippocampus in adult rats was actually making new neurons. What was at the time a totally fancy cutting edge technique. And what Altman told the world was, actually there is adult neurogenesis and it occurs in the hippocampus, and no one believed him. And most significantly among the people who didn't believe him was a guy who, while still young in his career, was already a grand old man, a guy at Yale named Pasco Rakich. And Rakich had done half of the textbook findings about how the brain wires up in the fetus, and totally amazing. And Rakich decided that is not possible. Altman must be doing something wrong. The adult brain doesn't make new neurons. It is not possible. And Rakich very, very assertively came down on the side of saying, this can't be for real. I don't believe this finding. And as the story goes, thanks to Rakich having a fair amount of influence at that point, Altman was denied tenure at MIT and spent the rest of his career at Purdue University. And a few years later, another researcher named Kaplan at the University of New Mexico was also getting some evidence for adult neurogenesis, and Rakich decided that could not be the case either. And famously in a conference, when Kaplan had presented some of his findings, famously said, well, you may think those are new neurons in New Mexico, but here in New Haven, those certainly do not count as new neurons. The whole field was crushed. Altman was forgotten, worked in obscurity forever after. Oh yeah, wasn't there some guy in the 60s who said that there's actually adult neurogenesis, whatever, I don't know, but nobody really picked up on that. The adult brain doesn't make new neurons. And then things started in the 1980s, this revolution of adult neurogenesis. And it started off inauspiciously because it started in the last place that would convince anyone, Rikic or otherwise, that something interesting was happening. Birds, bird brains. Think about it. There are bird species where every year they learn a new song, a new mating song, a new territorial song, whatever. And what was found was birds each year, for making new songs, they make new neurons. In the bird equivalent of the hippocampus, this was a guy named Fernando Nottebohm, senior figure in neuroethology at Rockefeller University, and his graduate student, Steve Goldman, who was a classmate of mine at the time. I was at Rockefeller, and they announced to the world with new improved techniques from the 1960s that birds make new neurons in adulthood. And in fact, if you stop that from happening, they don't learn new seasonal songs. Whoa, totally cool, amazing. And everyone said, well, that's nice. That's nice for Fernando and his little birdies. But that's got nothing to do with real animals, and certainly not with humans. And it got kind of ignored, except that it was lurking on the fringes. And in the subsequent years, more and more findings emerged from two places. Most dramatically, one was from the Salk Institute, actually UCSD at the time, and later the Salk Institute. Fred Gage, known as Rusty Gage to everyone. Rusty Gage is one of the most renowned, most respected neuroscientists, and he was getting evidence there at the time that you see adult neurogenesis in mammalian brains. Meanwhile, the same was going on at Rockefeller. A pair of graduate students working in the lab of someone named Bruce McEwen. This pair, someone named Liz Gould and Heather Cameron, and they were getting evidence for adult neurogenesis as well. And this was very exciting to me because that's the lab I had done my PhD in about 5 years earlier with Bruce McEwen, one of the gods, and Gould and Cameron were showing adult neurogenesis. And nobody believed them or Gage. There was tremendous resistance, with Rikic again leading the charge, and Rikic, who was particularly hostile. It was noted when it emerged eventually that Gage at the Salk Institute and Gould at Princeton, they really became the two architects of this revisionism of adult neurogenesis, that Rikic was really aggressive and really unpleasant with Gould. And I think there are good reasons to think that that would not be the case if Gould had had one less X chromosome. And that's always been part of the whole epic story of it. But nonetheless, they were getting more and more evidence for it with new cool techniques. And there was this rear guard resistance. Okay, Rikic and colleagues would say, yeah, you're seeing new neurons, but there's not a lot of them. There's not enough for it to be interesting. Okay, yeah, it's a lot of them, but that's in rats. You don't see them in monkeys. Okay, you see them in monkeys, but not in humans. Classic work, Gage and eventually Gould showing it occurs in humans. And then the last ditch attempt was, okay, so the adult human brain makes new neurons, but they don't do anything. They're not interesting. They're not functional. But slowly the tide turned, and by the mid-1990s or so, this revolution. Adult neurogenesis occurs in the mammalian brain. And at that point, people began to get more insights into the subtleties of this. And again, Liz Gould and Rusty Gage were sort of the main motors behind this, but all sorts of interesting stuff. First, you did not see neurogenesis all over the brain in adults, only in two areas. The first one was Altman in the 1960s redux in the hippocampus. And the second was this little pocket of neural stem cells near the olfactory bulb. What is that about? These are the two places, new neurons being made in the adult hippocampus and in the olfactory bulb. And 99% of people ignore the olfactory bulb part because, like, that's just weird, but hippocampus was the center of the universe, and people began to first focus on, so what kind of things stimulate the birth of new neurons in the hippocampus? And Gage and Gould and others eventually started showing cool stuff like exercise, mental stimulation, cognitive stimulation, really interestingly, estrogen. All of these were boosting up neurogenesis in the hippocampus. What sort of things were suppressing neurogenesis? And that made perfect sense as well. Stress hormones, brain inflammation, all sorts of things that were already known to be bad. You could modulate rates of neurogenesis. You could boost it up in a stimulating and rich environment. You drove it down with stress, and this was making wonderful sense. And people slowly began to figure out what these new neurons in the hippocampus were doing. And it's a term that's now used, pattern separation. Okay, you have learned there's these creatures that walk around in the Galapagos or swim around in Finding Nemo, and they've got a shell on their back, and you learn those are turtles, and your hippocampus helps you with that. But then you learn to modify that fact. You learn actually there's turtles and tortoises. You have to refine turtle-y things to separating between these two categories. That's what the new neurons were good for. Pattern separation, something that you knew already, and now you learned, oh, there's actually dolphins and porpoises, and there's rabbits and hares, and there's, when you get some fine tuning of it, that's what the new neurons were for. Incredibly cool, exciting stuff. Meanwhile, the olfactory bulb story, virtually no one was paying attention to this until there was this bombshell of a finding that I loved, which it turns out, if you're a female rat and you get pregnant, you get rid of all of your neurons in your olfactory system and you replace them with spanking new neurons. You boost up neurogenesis like crazy in the olfactory system. People showed this was caused by prolactin, a hormone that's elevated during pregnancy. And whoa, and by the time you give birth, you've got this whole renovated, brand new olfactory system. What was that good for? You go in and you block that neurogenesis from occurring in the olfactory system of a pregnant female rat. You keep it from happening, and what happens is she never learns to recognize the smell of her babies. You need it for bonding. And that's totally cool also. And there's some evidence that the same is occurring in humans, and human olfactory imprinting on their babies goes on. So here's where the field is at. It's totally great. It's totally wonderful. What about Altman? Because this is now the hottest topic in neuroscience. If you're not doing adult neurogenesis, you were mocked out of the club. And what about Altman? And Altman's sitting there, and he's an old man by then, and he says, you know, it's nice that people believe it. I knew what I was seeing. He was not particularly bitter over the course of it. He was not particularly triumphant at this point. All sorts of folks kept nominating him very appropriately, I think, for a Nobel Prize, and he didn't live long enough to get it, but I thought that was a grave omission. And he had this remarkably sane, level-headed view about all of it. Altman, a refugee Hungarian Jew, he said, I survived the Nazis. Like, whether people believe what I'm finding with neurons or not, you know, you put stuff in perspective, whatever. And he died a few years ago at age 90. So that's the whole field. That's the story. Until a few years ago, the whole field gets tipped on its head. And this was a finding published in the journal Nature, which is the fanciest, most prestigious science journal on earth. In Nature, a group from UCSF, University of California, San Francisco, and a lab headed by someone named Arturo Alvarez Bulla, who was also a classmate of mine in grad school, who headed this whole large lab on development of new neurons and how things wired up. So Arturo and the lead researcher on it, a postdoc of his named Sean Sarles, were doing the most thorough study of adult neurogenesis to date with human brains and monkey brains and rodent brains from fetal life throughout the lifespan. They had a bigger sample size than all the previous human studies put together, and they had state-of-the-art 2015, 2016 techniques that were much better than the ones in the 1990s, which were much better than Altman's ones in the 1960s, far more sensitive techniques, this technical tour de force. And they found something really disturbing. You look at rats, and yeah, they make new neurons in the hippocampus in adulthood. You look at monkeys, they do the same thing. You look at humans, and by adolescence, you don't see any new neurons. The things that were being identified as new neurons from the earlier generation studies turning out to be a different cell type because people were getting knowledgeable enough to distinguish it between, whoa, rats, monkeys, yay, revolutionary. The adult human brain doesn't make new neurons. And as you can guess, this generated a furor like you can't believe. My personal view is that was a phenomenally carefully done study. And its basic finding with these new techniques has since been replicated. But just for objectivity, I also have to say that Sean Sarles, the first author on that paper, was my grad student. He was one of my all-time star grad students. And when he finished, he went on to this lab at UCSF. He now has his own lab at University of Pittsburgh, where he's a professor. So I'm a little bit biased, perhaps, but we've got this controversy. And suddenly, the revolutionaries from the 1980s and the 1990s are the ones saying, no, you don't know what you're doing. Of course, there's adult neurons there. And Rakich, who is still on the scene, Rakich, who in the 1990s finally admitted that there are new neurons in the brains of rodents and monkeys because he could show it in his own lab. So he finally believed it. And now, years later, Rakich, who is still running a very productive lab, Rakich also confirms that with these new standards and more stringent techniques, you don't see new neurons in the human brain after adolescence. So there's this gigantic war going on right now where the irony is what used to be the Young Turk revolutionaries are now the old guard with everything to lose from this new revisionist finding and all sorts of bad feelings, and everyone is convinced the other doesn't know what they're doing. And amid that, there's little hints of something even more interesting. Oh my God, you're saying a rat brain, a rat could make new neurons when it's in an enriched environment? A monkey can do it, but a human can't? That seems so illogical. There's coming the first hints now that what humans are doing when they need a new neuron online is actually much more elegant, but that's just a very slowly emerging story. Okay, so neurogenesis couldn't be revolution, acceptance. It's not quite what the story was. The usual arc of science finding out new cool stuff.
Okay, so Matthew from New York asks, as society becomes more complex and develops, and even potentially incorporates a reformed quarantine model of social justice without free will, how do science and religion reconcile its differences?
Well, my very dogmatic, hard-assed answer is you don't reconcile the differences. They cannot be reconciled. So what do we got here? We got, harking back a couple of weeks, incredible heterogeneity of religions. Let's restrict our discussion here only to religions that have what have been termed big gods. Gods who monitor human behavior, gods who know what you're up to, gods who judge you. So, is modern science reconcilable with this notion of God as an active judgmental agent in things? And I think no way whatsoever. And the main sort of cleanest way of stating that, this wonderful quote from Jerry Coyne, who's a renowned evolutionary biologist at the University of Chicago, and I'm not going to have the exact quote here, so I'm paraphrasing him saying, In religion, faith is a virtue. In science, faith is a sin. The entire backbone of the scientific venture is falsifiability. Whatever it is you think you know about how the world works, you are willing to give that up in the face of evidence that your view was wrong. Falsifiability. And you could not get more of a contrast. The whole point of religion is faith. If you want God to show that he's there, Jesus jumping off the temple in Jerusalem, Satan daring him to do that. Oh yeah, you're the Son of God. Jump off the temple and see if God catches you. And Jesus saying, no, I'm not going to do that, because what if God shows his hand? What use is religion? The whole point is you don't know. You have to have faith. Religion is all about the unprovable. Science is all about rejecting what you think is currently reality when it is shown to be false, falsifiability. So in principle, a priori, the two are completely incompatible. Nonetheless, people have tried to find meeting grounds between science and religion. One comes from the Harvard geologist Stephen Jay Gould, the second Gould of this episode. Stephen Jay Gould, who late in his career, when I'm not sure what was going on with him in terms of his religious stance, a strident Marxist atheist earlier in his career, back many episodes, one of the strongest critics of sociobiology, Gould came up with this concept that he called non-overlapping magisteria. It's not really a fight. Science and religion are relevant to different things. They're not overlapping, and they could both be right because they don't have the same concerns. If you want to go look at a gazelle and you have a scientific view and you want to understand the biomechanics of how it leaps and is so graceful looking, that's great. And if the person next to you wants to look at a gazelle and see God's grace and this beautiful animal and a sense of awe and humility, yeah, go for both. You can listen to Bach and you can have religious feelings from it. You can listen to Bach and understand the structure of the fugues. They don't have to be in conflict. Can't we all get along? The notion of non-overlapping magisteria. There's one area where people do that pretty regularly, and that's cosmologists, physicists. And when you look at the rates of belief in God among different groups of scientists, you see the highest rates among physicists and cosmologists. These are the folks who are sitting and trying to figure out if God rolls dice or not, starting with Einstein. When did the universe begin? Was there a beginning? What was there before? What happens after? All of those big issues. And an awful lot of cosmologists thinking about these massive and many ways ultimately unknowable, gigantic questions, find that is completely compatible with the notion of there being a God, even a God who is involved in human affairs. So relatively speaking, there's a lot of folks out there who are reconciling their being physicists, cosmologists, with religiosity. Then there's a realm of science, the life sciences, where one would think there is no way to reconcile the two. If as a scientist, as a clinician, you're spending your life doing pediatric oncology, you're spending your life telling parents that their 3-year-old has 6 months to live. If you are doing geriatric dementia, if you do any of these areas where you look at it and as a scientist, as a clinician, you are imparting this news, and it had always struck me as inconceivable that you could do a job like that day in and day out and continue to believe that there is an omnipotent being. And mommy, back in her clinical psychology, neuropsych days, working with dementia patients, that sure bolstered her sense there can't be a God. How can there be a God who gives cancer to 3-year-olds, who allow the Holocaust to happen, etc., etc.? So that seems completely incompatible. Yet, there's all sorts of people in those branches of science who are capable of being religious, find it being compatible, and this is the familiar realm. God works in mysterious ways. God is giving you a challenge to see if you're up to it. God is granting you the grace of showing the resilience to put up to this challenge. God can do anything that is doable. And there happens to be some things that are not doable, like preventing your child from getting all sorts of ways in which people, a subset of people who are spending their time watching children dying of cancer, somehow reconcile this with there still being a God. The domain of science, of life sciences, which I think is absolutely irreconcilable, as soon as you are getting into the science of trying to understand why people do what they do. Try to understand if people have responsibility for their actions. Try to understand if things like blame and punishment and praise and reward make any sense whatsoever. If a notion of justice being served, if a notion of somebody being entitled to a happy, healthy life or being entitled to a life spent behind bars, any of those, as soon as you are getting into the realm of the science of understanding why people do what they do, it is absolutely, utterly, implicitly, constitutionally, fundamentally, foundationally, all of that incompatible with the notion that there is a God who has given us free will. And because of giving us free will, he then has the audacity to judge us afterward for what we did with our free will. When it's getting to that realm, I do not see any possibility of reconciling the two.
Finally, Stephen from Seattle says, humans are born with the least developed brains of all primates. We take an enormous amount of time to mature into independent, functioning creatures. Why are we so dependent on others to survive?
Well, because we insist on being a fancy, complicated species. Old terms in ecology, the contrast between what were called R-selected species and K-selected species. Long history of that, island biogeography, whatever. It's a broad contrast between two different types of species, two different life histories. R-selected species, you're not going to live for long. You give birth to a whole lot of kids. You do minimal parental care. They're off on their own. Good luck to them. Most of them don't make it. And it's a flood the market, quantity over quality approach. In contrast, you get K-selective species, long development times, long lifespans, long periods of dependence on maternal care. You give birth to one or two at a time, not hundreds, and quality over quantity. Classic quantity over quality salmon. You just lay a million eggs. Most of them don't make it, and mom is long gone. And in contrast, primates are the most K-selected of species out there. These days, it's more likely called a fast life history strategy versus a slow life history one. All right, so primates, in contrast to salmon, you're only giving birth to one or two kids at a time, and it takes them a long time to mature, and you invest a ton of parental, maternal energy in raising them, and you've got a quality strategy. And among primates, you see this dramatically, and you have a contrast, forget with salmon, with lots of other mammals out there. A mama giraffe gives birth, and the baby giraffe plops out and falls 6 feet on its head and gets up and dusts itself off, and it's often trotting around soon afterward. It is, jargon in the field, highly precocious. In contrast, primates are dependent. They're useless for weeks, for months afterward. They're dependent on mom. $10 fancy term in the business. In contrast between being a precocial species, their term being an altricial species. Primates are altricial. Slow maturation, long dependence on mom, enormous amounts of maternal care, maternal education that goes into raising them, and a higher likelihood of any given kid surviving. So primates are highly altricial, long periods of dependency. Now, among the primates, humans are the most altricial, which is to say, we are the most useless, helpless, and unable to do anything at birth of all the primates. Other primates, they're wobbling around and walking within a few days. We have much delayed And the other is that it takes a larger percentage of our lifetimes for our brain to fully mature into adult levels of function. So, why are human brains more altricial, more dependent on slow maturation, more dependent on mom investing energy in your successful slow maturation than any other primate? Two areas where answers come in. The first one is one very yoked to gestation. You look all across the primate species and you look at the relationship between how long a primate species lives and how long its gestation period is. And there's a very clear relationship between the two. And you extrapolate on this curve and you then look at humans, and based on how long we live, human pregnancy is supposed to be, I don't know, 14, 15 months long, much longer. By the standards of all the primate species, we are born prematurely. How come? There was always the classic explanation in the field, which is we humans with our big heads, you got to give birth prematurely, 9 months representing premature by primate standards, because humans with these big heads, when the third trimester fetuses, you better give birth to them sooner or else their head's going to be too wide to fit between within the pelvic canal to go through there, and that's a disaster. And there was all sorts of evidence suggesting that head size, brain size was a trigger for the onset of labor. What people have learned since then, something about lung maturation in the fetus is also a signal for when labor should begin. There's another body of evidence, mostly this work of this anthropologist named Peter Ellison at Harvard, saying that what you get at some point is like a human fetus is so expensive with this big brain stuff. Not only at some point does the brain and the head get in danger of not being able to fit through anymore, at some point the fetus is just too expensive to mom to be feeding intrauterинely, and the beginning of an energy shortage in the fetus generates a signal, time to give birth. All right, so we've got that world where humans by primate standards are born about 5 or 6 months prematurely. So we gotta mature past that stage. But then we get into the second sense of humans being an altricial species, which is we got parts of the brain that are not fully mature until you're a quarter century old. The prefrontal cortex, part of what makes it the greatest part of the brain. We've talked about endlessly what the prefrontal cortex is about, doing the harder thing when it's the right thing to do. Prefrontal cortex, delayed maturation of it in all the primates. And all the primates is the last part of the brain to fully mature. But in us, by miles, it is delayed. In all the primates, it's serving that same purpose. What's the prefrontal cortex for? Whether you're a marmoset or a human, any of the primates, it's a very fancy part of the brain that does gratification postponement and long-term planning and impulse control and emotional regulation. And that's important for any primate, which is why it takes longer to develop than in a gopher or a rat or some such thing, because primate sociality has a lot of challenge. It takes a long time for any old primate to learn how to do the right thing, what counts as the right thing. And for other primate species, frontal cortical maturation is mostly about it's going to take a long time to get the social experience to be good at game theory, to understand dominance hierarchies, to understand affiliations, to understand when it makes sense to cooperate, when it makes sense to cheat, when it makes sense to optimize by stabbing your coalitional partner in the back. A lot of what the delayed prefrontal cortical maturation in all the primates out there is just getting good at social strategizing. And no genes can specify that. You got to have some of the things that primates are really good at, which is observational learning, social learning, and a long learning period where your brain is still being wired up in this very important area. You get to humans, and obviously delayed frontal cortical maturation is very important for learning by experience what game theory strategies, when to cooperate, when to cheat, all of that. With humans, we've got an additional thing that no other primate does out there, which is we have to learn the culture-specific hypocrisies. the culture-specific rationalizations. Thou shalt not kill. However, if you kill them, we're going to give you a medal. Whoa, that can cross the wires of a plain old rhesus monkey. Doesn't that seem contradictory? Know how to learn. Don't lie. Don't lie. But lie if grandma has brought you a present that you already have. Don't tell her you already have it. Wait, that's lying. No, that's an okay lie. To the adult version of, oh, are you sheltering refugees in your barn who are trying to ethnically cleanse? Nope, haven't seen any of them, you say while they're back there in the hay. Yeah, context, situational ethics, rationalizations, that's really complicated stuff. The difference between do as I say, not as I do, the difference between the spirit of the law and the letter of the law, primates learning when to cheat at their games of social strategy, that's pretty challenging. Learning when human hypocrisy has its place, that is so much more complicated. That's why I think we spend a quarter century having the final part of our brain, the prefrontal cortex, mature way longer proportionally than in any other primate because we're doing real fancy things with it. Of course, we get a horse and cart chicken and egg cause and effect thing there. Did we develop delayed frontal cortical maturation because we had to get this social expertise at rationalization and spirit and letter of the law, all of that? Or was the social selection for being good at that the driving force on delayed brain maturation so you get really an adept frontal cortex, obviously each feeding on the other?
And that's episode 47. Keep submitting your questions at the form found in the Instagram story highlight and bio or the YouTube video description. I'm Offspring Sapolsky, and thanks for your continued support of Science and the Beard.